Hello, welcome to Ilma. In the last class we had done and started a new subject, biology. In the last class we had started with structure of chromosome and cell cycle and division. In that chapter we did the portion of chromosome within the cell, and in today's class we will be continuing with the same chapter, and we will be focusing basically on the cell cycle part of the chapter, not the cell division. In today's class we will be focusing on cell cycle, so let's begin with that. Now, firstly we need to understand what is the need for a cell to divide. It is needed for growth, replacement, repair, and reproduction. Now for growth, as for example, for a child grows, his body elongates, not taking the cases of dwarfism or all that. A normal child, a body elongates, it broadens, and the size will increase. For a plant or animal, anything, the size will increase. Now that cell increase is by the cell division, which is termed as growth of the organism. From one cell it will become 2, from 2 to 4, 4 to 8, 8 to 16, like that. That way the size increases. Like from the zygote we get a complete human being in the adult form. Now that is an example for growth. Now cell is also required for replacement of worn out or dead cells. In the normal functioning of the body, millions of cells die every second, and so to keep up with them and not to have any lack of cells for functioning, we need to get new cells regularly at a very instantaneous basis. Therefore we also divide, the cell division takes place to create many millions of other new cells, which would take up the place of that old cells and would carry on their functions respectively. For example, the skin cells are constantly, the skin layer is just a layer of dead cells, which is also known as the sclera. They are just flattened dead cells, which would if we wash our hands, we lose many skin cells, uncountable type, and many skin cells are lost if we wash our hands or in that way. And this skin cells or skin layer will be replaced by the layer between our skin, which is the endodermis, which will produce more cells, and as the cells grow up they will die and form a layer, flattened layer. That will be in the skin chapter of class 9. We must have studied it. Now coming to the repair part, if our body gets some damage from anywhere, for example we may cut a cut in our body, the blood is lost, might be very little but still to blood is lost, and that blood might contain lots of RBCs and WBCs. WBCs will be less in number than the RBCs, but RBCs will be lost. That will be supported by the bone marrow, which will produce the RBCs within the marrow. That's why the bone has so vascularity. That means so many blood vessels enter and leave the bone, because they will provide nutrition to the bone marrow for producing new RBCs. And that's in some bone marrows they are just produced, in the others they are matured. Now that's for the repair part. Like if we get a cut on the hand, the skin cells will also need to be repaired. If our blood vessels are leaked, that means absolutely the endodermis is hurt and it needs to be repaired by new cells. That is for repairing. Now we come to reproduction part. Now there are two types of reproduction: asexual and sexual reproduction. That would be, suppose in an organism like microorganism, amoeba or somewhat like that, they would divide, or bacteria, bacillus, they would divide by binary fission. Or in fungi or cells, organisms other than that, they would divide by budding. Yeast is by budding. Then there is fragmentation of Spirogyra, Chlamydomonas. These microorganisms mostly carry out sexual reproduction. There are some higher organisms also, but these are the basic examples. Binary fission for bacteria, then spore formation for bacteria. That is not for reproductive reasons, that is just for protective reason. Spore formation is not that. For fungi, spore formation is a method of reproduction. Now in sexual reproduction there will be meiosis. Meiosis is a term. In this chapter it will be studied in greater details. There are two terms, mitosis and meiosis. Mitosis is the normal cell division in any part of our body, and it will be just like the binary fission type, but in much more complex way. The cell will be divided into two daughter cells in many steps, and in meiosis there is also similar to mitosis but not the same as mitosis. In this, suppose there is a cell, it will divide into two cells. Now those will divide into two other cells. Now this is done by the pathway of mitosis, but this is not done by the pathway of mitosis. I mean, these will be haploid in number, n number of chromosomes, and the normal cells we have in our body are diploid, which will have 46 chromosomes or 23 pairs of chromosomes. This four cells will be haploid, having only 23 chromosomes. And in this there's a difference that in mitosis there are only two daughter cells. In meiosis, the complete process, there are four daughter cells. Now we will be going in further details in the next classes on this chapter. Now let's begin with the main topic of today's class, cell cycle. Now a cell cycle is the lifespan of a cell. It means it grows starting from a daughter cells, then it grows and then it becomes adult, then it will divide that is the complete cell cycle. Now in that there are there are two types of firstly, the first part of cell cycle is the interphase. Interphase comes just before mitosis, which is the actual division. In this phase, the cell prepares for mitosis, which means the cell has to get big enough in size such that if the cell divides, the daughter cell, which will not be too small and will cease to exist, then therefore it should grow bigger than normal size, then it will divide in two. In interphase there is DNA forming, means DNA replication. That is why a diploid number is maintained, or else it will lead to mutations or meiosis in similar ways. Now in this process, the DNA material, ribosomes and proteins are multiplied and replicated for the further growth and division of the cell and into the daughter cells, and these daughter cells will use the ribosomes and proteins which are divided to grow faster into adults. Now in this phase there are three further cells, minute phases. They are G1 or growth 1 or gap 1 phase, G2 there will be S phase, which is the synthesis phase, and then the G2 phase, which is the gap 2 or growth 2 phase. There is another phase in between, which is the R phase, also known as the resting phase. This is a separate, this will not be if these three phases are being followed by the cell. Now in growth 1 phase, the basic function of growth 1 phase is to divide the proteins and multiply the proteins, ribosomes, and then prepare it for the synthesis phase. In synthesis phase there is DNA replication, therefore the ribosomes needs to be greater in number, they will help in the DNA replication, and so for the proteins they will be also utilized during the DNA replication. Now in synthesis phase, as I said, synthesis phase is for synthesis of DNA. The DNA will be doubled. Like in the previous class, I had used the word duplicated chromosome. This is the place where the DNA will be doubled. It will not be formed into a chromosome. The chromatin will only double itself, and then it will stay like that. In the mitosis period, the DNA will thicken and form chromosomes after the prophase or at the end of the prophase. Now, in G2, gap 2 or growth 2 phase, this is the phase in which the protein and ribosomes again are further synthesized for the division of the cell to take place, such that the daughter cells don't lack anything in their growth. And again in G1 phase, the organelles, some organelles like mitochondria and chloroplast, mitochondria in both animals and plants, and chloroplast in plants only. Chloroplast will help in photosynthesis. These two organelles have separate DNA, not the same DNA as the cell. This will have separate set of DNA, therefore it can divide on itself. Whenever there is a lack of or more pressure is being applied on the cell, more oxygen is needed, the mitochondria will be dividing, or there more photosynthesis is required for the plant, the chloroplast will be dividing. So that's how the growth 1 synthesis and growth 2 or gap 1 synthesis and gap 2 phase works. Now coming to the resting phase. After the growth 1 phase, the cell which is the parent cell might enter into the resting phase. This will start from here. This will not be executed. These two phases will not be executed if this phase is being executed. Now in the resting phase, it is the decision of the cell whether it wants to divide further or whether it would like to stop right there, and it is again its decision whether after entering the resting phase it would want to leave the resting phase and start dividing again if the body commands it, and or it should stay in the resting phase and no more divide further. Now in some cells like nerve cells, brain cells, heart cells, they do not divide any further. They will be the same constant number until they are grown. They might just grow in length and size, but they will not divide, as the nerve cells have lost their ability to divide. The heart cells are the same from the beginning to the end. They will just grow in number. After they are born, they will not be dividing. Now, as cardiac cells never rest, they are the heart cells, muscle cells, heart. Now, there are some cells like liver cells which divide the frequency of division is very low. They will divide in very short, long intervals of time. I mean, if the DNA cells are needed, they will divide in 2 to 3 years, once in 2 to 3 years. And if there is some urgent need for that, they would still take some time for division. They are not dividing so easily. Now that's why we get liver problems much easily, because if the liver cells are destroyed, we would not be getting the destroyed liver cells back in a very short period of time, and we would need the medical help for that. Now the resting phase is discussed. We will be going now in the resting phase. Yes, the cell cycle is further having more parts. Mitosis is one part of the cell cycle, and instead of this mitosis, it might have a meiosis. Meiosis will be discussed later. Now mitosis is again having two parts, one major part and one minor part. Karyokinesis and cytokinesis. Now, as the name suggests, caryo like eukaryotes and prokaryotes we have studied. we have studied. Eukaryotes means modified new type of nucleus, and prokaryotes mean primitive type of nucleus. In microorganisms, these are studies. Now karyo, like in eukaryotes, karyo means nucleus, and kinesis means division. Therefore, this means division of nucleus. And cyto similarly comes from the word cytoplasm, division of the cytoplasm. Now cytokinesis, cytoplasm division of the cytoplasm, or we can say it as division of the complete cell. Now between these two stages, there is a stage where the cell would have two nucleus. At this stage, there would be a small furrow. This will be explained in the telophase of mitosis in the next class. This furrow will deepen and deepen and deepen, and finally this furrow will reach each other, and the cell will completely divide into two daughter cells. Now after karyokinesis, only cytokinesis can lead to two daughter cells, or else it will lead to a cell having two nucleus. This is just after karyokinesis, and this will be after cytokinesis. So cytokinesis is a very short process. It will not take much time. Karyokinesis is a lengthy process, but still too short in time. Actually, it is still short, but in comparison to cytokinesis, it is a lengthy process, having prophase, anaphase, metaphase, telophase, all 5 stages. Now we will be going to meiosis. Now meiosis is a short introduction to meiosis. The greater details will be done in the next few classes. Now meiosis, as I said, one cell divides into two cells. Now those will divide into two cells. This is for now for ICSC. This is not required to study how the meiosis is taking place. We just need to study what is the properties of meiosis, why it is done, and all that. Now these two cells will have some genetic differences. The genes will be the same. In total, the genes will be the same, but each chromosome, duplicated chromosome, will have some differences, which would be from the other chromosomes. For example, in a male, it would be X and Y chromosomes in humans. Now some part of one X chromosome and the other, this is for the autosomes, will be exchanged. Like this part shall be exchanged with this part. Therefore this chromosome will look different, a little different from the previous X and X chromosomes, autosomes. Now this will be again discussed. This is just a short introduction on cell cycle. Now there's a chromosomal genetic interchange, and we will be studying about the chiasma and crossing over process. This is just for genetic variations, and this eventually will lead to mutations and evolutions by asexual reproduction, unless there is some type of mutation to the genes by some radiation or some mutators. It will not lead to the evolution. It will be the same as the it has been from the beginning of the cell. It will be not evolving. There needs to be some mutation in that. But in this type of genes, slowly in a lot of time, many years, they will start looking different. Like the same man which has come from here, which is said to become from our ape, will look like Homo neanderthalensis, Homo sapiens. All that differences will be made by this. So this would be the end of today's class. I had completed this cell cycle topic. Mitosis and meiosis was just an introduction of the next topic we are going to do. Now there might be a few more classes. There will be actually a few more classes of this same chapter. And if you like the video, please do like, share, comment, and subscribe. And You may WhatsApp me at 9874944154 or comment in the comment section for any queries or any information you require. And you may also ask your doubts in the comments or in the WhatsApp number. So that's all for today. Thank you.